Hello viewers. First of all, thank you for coming back to this channel. Today we will discuss about basic structure of immunoglobulins. You know that antibodies are nothing but immunoglobulins. The the protein model that you are seeing on your screen is an antibody. Yeah, true. This protein model or antibody model is famous as Angel of the West, and this is installed at the Scripps Research Institute, Florida. Before I go forward, I would like to tell you a bit about electrophoretic analysis of serum proteins. It was way back in 1939 when Tiselius and Cabot did certain experiments, and they found out that the antibody molecules are gamma globulins. What they did was they immunized rabbit with ova albumin and then analyzed the serum. Okay, so this analysis of serum just after immunization was serum, you can say untreated serum. And in the second sample, they treated this serum or mixed this serum with the antigen itself, that is ova albumin. And then they removed the complex formed. And the leftover proteins, serum proteins, were analyzed on the gel. And they saw four major peaks on the gel. We can say four major bands on the gel, and their respective concentration is also shown here in terms of absorbance. Right, so they saw a peak for albumin and peak for alpha globulin, beta globulin, as well as gamma globulin when it was untreated sample. When the treated sample was run, where antigen antibody complex, I would say that and it was antigen antibody complex which was separated because a sharp decline in the gamma globulin peak was observed by them. And through this study, they concluded that okay, this gamma globulins are nothing but immunoglobulins or the molecules which are capable of interacting with the ova albumin. That's why they got separated during the in the treated sample, that is with the antigen. Okay, so through this experiment, it also can be concluded that antibodies are serum gamma globulins. Okay, Tiselius was awarded with the Nobel Prize in Chemistry in 1948 for his research on electrophoresis and adsorption analysis, especially for his discoveries concerning the complex nature of serum protein. Yes, he with along with the Cabot found out that there are different serum proteins and immunoglobulins are gamma globulins. So did a fantastic work and got the Nobel for his significant contributions. Now we will look at the structure of the immunoglobulin. Immunoglobulins, that is antibodies, are proteins, right? And they are not simple proteins, rather they are conjugated proteins. And that too they are conjugated with carbohydrates. That's why they are glycoproteins. Now let's focus on the antibody molecule. Here in your on your screen you are seeing an antibody molecule where these boxed structures are nothing but polypeptide chains present in the glycoprotein antibody. Okay, so here you are seeing one chain smaller in length and the second chain which is longer in length, and a copy of the same chains is present. Okay, this is the long chain and this is the smaller chain. So you are seeing here if this complete molecule is divided into halves, it will be a dimer of these two halves, right? And this half itself is a heterodimer of two polypeptide chains. So we can say that the antibody molecule is a dimer of heterodimer, right? A light chain is there, smaller chain is there, and heavy chain is there, right? And you know that polypeptides have C terminus and N terminus. In case of immunoglobulins or antibodies, all the N termini are facing towards one side, while all the termini are facing at the other side. Now, another feature of these antibody molecules are that they have immunoglobulin domains. Now, in this light chain, you are seeing two boxes, 1 and 2. So, these are nothing but immunoglobulin domains. So, light chain has two immunoglobulin domain, while in heavy chain, we can see here four boxes. So, four immunoglobulin domains are there. And you can also see that there are intra-chain disulfide bonds in each immunoglobulin domain, okay, both in case of light chain and heavy chain. And we can also easily see that intra-chain disulfide bonds holding these chains together. This light chain is held together with the this heavy chain with this interchain disulfide bond, while these two heavy chains are held together by uh, disulfide bonds in this particular area, okay, which is not uh, area having the immunoglobulin domain. This specific region, which is allowing the disulfide bond formation, is called hinge region. This hinge region is of specific significance because it is a cysteine rich and proline rich region in the heavy chain of the immunoglobulin or the heavy chain of the antibody molecule. Now, the cysteine residues that are present helps in formation of disulfide bonds, while the proline residue help in the movement of these arms of the antibody molecules in their environment for the catching of the antigens. Now, another this CHO group, which is depicted here, shows the presence of the carbohydrate. Okay, so the antibody molecules are dimers of heterodimers. They are made up of immunoglobulin folds. Their chains are made up of immunoglobulin folds. They are held together with the disulfide bonds. They are conjugated with carbohydrates. Right, these are specific features. Now you see that these immunoglobulin domains are named. There are some symbols written here, right? It's a customary way to depict the different regions of an immunoglobulin molecule, right? This is an immunoglobulin molecule. The region at the N terminus forms the antigen binding domain, or the antigen bind contributes to the antigen binding sites. Okay, and this is called as a variable region. These single immunoglobulin domains, both in light and heavy chain, constitutes the antigen binding domain. And this is the other name of this domain is the variable domain. So it is customary to write or to give it a symbol of V. V stands for variable. Variable region of heavy chain and variable region of light chain. Likewise, in this for heavy chain also and light chain also, we have a variable region in the heavy chain and variable region in the light chain. And rest of the portion of the antibody molecule is known as constant region. And all the immunoglobulin folds are given the code C. C stands for constant. Okay, all the immunoglobulin domains in the constant region are named as C, and if it's a constant region of the light chain, then Cl. If it's a constant region in the heavy chain, then Ch. And there are, because there are different immunoglobulin domains, so constant domain in the heavy chain and first immunoglobulin domain, second immunoglobulin domain, third immunoglobulin domain, right? And I will now would like to proceed to tell you that there are different types of or different classes of antibody molecules. In our body, we have five types of antibody molecules, or we have five classes or five types of antibodies: IgG, M, A, D, E. Okay, in these immunoglobulins or in these antibodies, only the heavy chain is different. So this classification or this of different antibody molecules is based on the type of heavy chain they have. Okay, in IgA, IgD and IgG, we have similar type of heavy chain. Okay, they have three immunoglobulin domains like shown in this figure, 1, 2 and 3. Okay, and in between CH1 and CH2 they have a hinge region. So three antibodies out of five, that is IgA, IgD and IgG, have a heavy chain that has three constant domains, and between first and second constant region of our constant immunoglobulin domain we have a hinge region. While in case of IgE and IgM, there are four immunoglobulin domain in the constant region, and there is absence of hinge region. There is no hinge region here, right? And instead of hinge region there is an extra immunoglobulin domain that functions analogously to the hinge region. And the heavy chains present in these five antibody molecules are named as gamma, mu, alpha, delta, and epsilon. So in IgG we have a gamma heavy chain, in IgM we have a mu heavy chain, in IgA we have alpha heavy chain, IgD delta heavy chain, IgE we have epsilon heavy chain. And IgA, D, and G have a different or similar type of heavy chain, while IgM and IgE have a similar heavy chain. Now, this variable region of the antibody molecule functions for binding to the antigen, cognate antigen, right? So there are paratopes that binds to the epitopes present on the antigens. So in the variable region, there are paratopes present which binds to the epitopes on the antigens, and rest of the biological functions or other biological functions are being carried out by the constant region. Okay, now the presence of carbohydrates helps in solubility of the antibodies, right? And these antibodies, these different types of antibodies differ in their not only in the heavy chain length but also their serum concentration, their half-life in serum in blood, right? So the type uh, also dictates the characteristics, concentration as well as stability, solubility, and half-life in the serum. So through that figure, you understood that there are five types of or five classes of antibody molecules, okay, having different heavy chains. Here you can see the difference very clearly. Each immunoglobulin domain is made up of about 110 amino acids. So if we talk about the constant region of gamma, delta, and alpha chains, okay, as there are three immunoglobulin folds are present, so its approximate length will be 330 amino acid, right? While in case of mu and epsilon chains, there is an extra immunoglobulin domain, and there is no hinge region. And we can easily calculate the average or approximate length of the constant region of these two heavy chains, mu and epsilon. It will be 440 because there are four in the constant region of the immunoglobulin. There are four immunoglobulin domains. Okay. I hope that the content presented in this video you could easily get it. And if you want to know more about immunoglobulins, please watch the next video on immunoglobulins. Thank you so much for watching this, and keep coming onto this channel for more videos, more updates, and more interesting content. Thank you.